So we shake our samples to homogenize them. That way any solids become suspended in the water column. And we add 10 milliliters of our tank water to each well. So while I'm shaking the next sample, I'm rinsing the pipette tip out. Because it's just tank water and our treatments are not different types of water, like clear water versus biofloc systems, we can use the same tip. If we had different types of water systems, we would want to use different tips for each tank. So this just saves us some work in the end. So I shake sample 5 while I'm rinsing the pipette tip. And then the pipette tip goes down into the water column and I draw up 10 milliliters. We pass 10 milliliters of tank water through each of the wells. And this is step 2 of total suspended solids and volatile suspended solids. Our filters have already been combusted and initial weights taken. And so we're going to repeat this process until we have all of our tanks into the millipore, all of our water samples, and then we'll turn on the vacuum pump and filter them. Okay, so at this point all of our sample water is in each of the wells of the millipore. I'm going to turn the vacuum pump on, and then you can see that the water begins to pass through the filters. And once all of the water passes through and the filters are dried, you can turn the pump off. If some sample wells are slow to filter and others are quick, we have stoppers that you can use to plug any well that has filtered to maintain that vacuum and allow the well that's slow to pass through. But you cannot alter the filter in any way when you're running TSS, VSS. You simply have to allow time in the pump. Sometimes if we're running biofloc systems or algae-based systems in the high tunnels, it could take an hour or longer to filter 10 milliliters of water. This particular trial, we are filtering very quickly and we're harvesting next week, so that's a good thing for us. So at this point, what we want to do is we'll take this nut off of the top. We have another step to take with our filters, so we can't disrupt them. So we have to grab both sides of the millipore, both plates, and lift it, and we'll set it aside. And from here you can take a look that each of our samples has been passed as it should be into our tube. We're going to use this filtered water to run all of our other water quality, ammonia, nitrite, nitrate, phosphate, alkalinity. Well, no, not alkalinity. That is done with non-filtered water. So I'm just going to put these in a test tube rack, and then we can begin our weekly water quality tests with these. Usually solids in system water will affect the results of the tests, and so we filter everything just as a preventative method. And we'll do so now, so I don't get my filters confused. Again, I'm going to grab both sides. I'm going to set this back down into the millipore. I'm going to stabilize this so my filters don't move while I tighten the nut on it again, because we have one more step to take with these. And then I'll take the second set of filters off. And we'll do the same. We're going to take these out. So you can see how that O-ring kicked out a little bit. You want to make sure that that's in the groove before you put the system back together so your vacuum is maintained. And so you can get a clip of the filters and how the color varies based on the amount of solids that are in the water. And I'm going to grab some distilled water. So our next step in this process is to pass 10 milliliters of distilled water through each of the filters. Grab a fresh, clean pipette tip. And so I'm going to take 10 mLs of distilled water, and I'm going to circle the edge of each well to rinse, to flush down any solids that may have attached to the side of the well. What this does is it's going to make sure that all of our solids get onto the filter, and also it'll rinse some of the salts through the filter, because we are running brackish water systems. And so when I'm doing this, I'm not being very forceful. I'm being gentle when I'm releasing the water so it doesn't create any kind of pressure that may dislodge any solid particles that are already on the filter. And then you can watch the filter. So a tip on basic pipetting: as you're drawing the sample up, run the pipette tip along the side of whatever vessel you're drawing it out of, and that will eliminate any water drops or accumulations on the outside of the pipette tip, which can also, your pipetting accuracy is improved if you're consistent in doing this. Not as important if you're passing 100 milliliters through, or you're diluting a sample, or many other different applications, but if you maintain the same accuracy and precision throughout your methods, your results will be more reliable. Okay, so I never lay the pipette down with a tip on it. I don't want any water going into the pipette, it'll ruin it. So I drop the tip in there. I'll turn this pump on, and you can see that water pass through. Everything filtered really easily, which doesn't always happen. Okay, so from this point, I'm gonna get the samples out of the way. So at this point, our filters are ready to go into the drying oven. And so I'm going to take the top off, and I'm going to ensure, if you look down there, you see that well number 11, the filter stuck. And so I'm going to hold that and just very lightly tap it to drop it down. And now we're going to make sure that we're starting with filter number 1. We always double check your samples. We're running 24 samples. We don't want to get them mixed up. And so I'm going to grab filter number 1, put it in crucible number 1. Sometimes pieces of the filter come off. You have to be very adamant about keeping everything together. So I want to make sure that that piece goes into crucible number 2. So sometimes if it sticks, well, this one is going to be a challenge. So sometimes you can attach it, but it's so far up, I'm just going to get it on the edge, and then it can adhere. So now I know that all of that filter passed through. Sometimes if the filters are hard to get off, you can release the hose from the pump or lift it up. That takes any vacuum pressure off. Or you can use these are millipore forceps. They're very flat on one end and very pointed on the corners. So you can use them to kind of get under the filter a little bit, to grab a hold of it. And so what I'm trying to do is stay on the rim. I'll show you on this one where there's more of a color. So I'm going to make sure that my forceps do not get into where the tank water has filtered. You see I'm on the outside rim where it's white, where there's no contact with tank water. So I'm going to try and maintain that so I don't contaminate between samples. So we're using a Quincy drying oven. It's a model 40 GC lab oven. All temperature gauges on all ovens may or may not be 100% accurate, so we flag where 100C actually is. You can see it's beyond that, so it just makes our life in the lab easier. I'm going to check our temperature gauge that we're at about 100 degrees, and we are, so we're ready to load the oven with our samples. And so when you're loading any oven, it's critical that all the samples get put in order. So I'm starting with 1 and loading the oven in increasing order of sample number as you read a book. So the first row will be 1 through 8, the second row 9 through 16, and so on. And the way we work in our lab is whoever loads the oven unloads the oven, because they tend to know the sample order and how they loaded it. And maybe somebody else has a different method that I do, and it just saves checking the crucibles to make sure everything is in consecutive order. So you can see I'm picking up 2 at a time. I would recommend starting with 1 crucible at a time until you're used to the methods, and then you can grab a couple if you need to. So you can see how they're loaded. Again, each of our crucibles has a number on the bottom that helps us keep track of what sample is where, just in case things get out of order. So now I'm going to close this oven, and I'm going to allow the temperature to get back up to 100 degrees centigrade. Once it gets to 100 degrees centigrade, we're going to dry the filters for 60 minutes, and then we'll put them back in the desiccator and let them sit overnight, and then tomorrow we will collect our dry weights.